Welcome to our overview of bioinformatic tools and how they shape biological research. We will move from core concepts to practical software and real-world applications. Think of this session as a roadmap for turning raw biological data into insight, with the title slide setting the stage for a guided tour through methods, platforms, and future directions. Before diving in, here is the journey ahead. We will introduce bioinformatics, spotlight major tools and software, and explore where they are used, from genomics to drug discovery. We will close by examining challenges and future trends. The circuit board image hints at the fusion of biology and computation that underpins everything we will discuss. What exactly is bioinformatics? It is the use of computational analysis to manage and interpret enormous biological datasets. The scope spans genomics and proteomics, studying genes and proteins for biological insight, and extends to systems biology, where networks and interactions come into focus. The small images echo these layers, from sequence patterns to interconnected systems. Why does this field matter today? Because modern biology produces vast data, and bioinformatics turns it into answers. From the bands visible beneath the petri dishes, imagine decoding patterns to reveal disease mechanisms. Those same interpretations drive drug development and enable personalized medicine by matching treatments to genetic profiles, accelerating discovery while improving care. Bioinformatics is inherently interdisciplinary. It blends biology, computer science, statistics, and mathematics to design algorithms that make sense of complex data. The hand sketching an orbital symbolizes this integration of disciplines. Just as important are software tools and databases, which standardize analyses and manage data so researchers can ask sharper questions and obtain reliable results. Sequence alignment sits at the heart of many analyses. Tools like BLAST and Clustal Omega compare DNA, RNA, or protein sequences to find similarities and differences. These comparisons uncover evolutionary relationships and guide functional annotation by linking unknown sequences to known genes or proteins. The helix image reminds us that structure begins with sequence. For genome and transcriptome studies, platforms such as Galaxy and IGV provide end-to-end workflows. They handle raw data, offer interactive visualization to interpret complex patterns, and streamline analyses for users with varied expertise. The laboratory scene with digital overlays conveys how these tools bring bench work and computation together to produce clear, reproducible insights. Understanding proteins requires seeing and predicting their shapes. PYMOL delivers detailed 3D visualization for exploring molecular interactions, while Swiss Model predicts structures computationally. Together they support function annotation and guide drug design by revealing binding pockets and interaction sites. The colorful particle field evokes the intricate, dynamic nature of protein architecture at atomic resolution. Genomics meets the clinic through personalized medicine. Bioinformatic pipelines analyze individual genomes to pinpoint variants linked to health and disease. With those insights, care teams can tailor therapies, adjust dosing, and avoid adverse effects. The gleaming DNA strand on this slide underscores how patient-level data becomes actionable when algorithms translate sequence into guidance. Drug discovery increasingly depends on computation. Algorithms rapidly propose viable targets, virtual screening filters vast compound libraries, and modeling predicts how molecules interact and what side effects might emerge. The interface showing past and warning statuses captures this funnel, where bioinformatics de-risks decisions long before expensive clinical trials begin, saving time and resources. Comparative and evolutionary analyses broaden our view across species. By comparing genomes, we detect similarities and differences, trace evolutionary relationships, and identify conserved elements that mark essential functions. The tree assembled from pencils symbolizes shared roots and branching paths of life, reminding us that cross-species clues often illuminate human biology most clearly. With the foundations laid, let's turn to the road ahead. The next section highlights the grand challenges facing bioinformatics and the trends reshaping it, from scaling to massive datasets to harnessing artificial intelligence responsibly. Consider this a pivot from what we can do today to what we must build for tomorrow. Biological data is exploding, and the binary rain in the image says it all. Managing this growth requires smarter storage, efficient compression, and cloud-ready architectures. Equally critical is overcoming computational bottlenecks with parallel processing, optimized algorithms, and hardware acceleration so analyses remain feasible as datasets stretch from gigabytes to petabytes. Artificial intelligence is weaving into every layer of analysis. Enhanced pattern recognition improves accuracy in noisy datasets, machine learning supports predictive models of disease and treatment response, and automation scales routine tasks. The montage of networks and robotics hints at this shift, where human insight is amplified by models trained on vast evidence. Power must be matched with responsibility. Safeguarding genetic privacy, obtaining meaningful informed consent, and ensuring responsible use of genomic data are non-negotiable. The lock, signature, and icons on this slide emphasize governance and trust. Ethical, legal, and social frameworks protect individuals while enabling research that benefits patients and communities. To conclude, bioinformatics is a dynamic engine for discovery. Its powerful tools transform raw sequences and signals into biological understanding, fueling advances in diagnostics, therapeutics, and personalized care. As we embrace AI and tackle big data challenges responsibly, we gain deeper insight into life's processes and move closer to truly data-driven healthcare.